Okay, so the DNA fingerprinting, 99.9% of the base sequence among humans is same. Okay, now DNA fingerprinting involves identifying differences. Identifying differences in some specific regions in DNA sequence called as repeated DNA. Now, because in these sequences, a small stretch of DNA is repeated many times. Now, this repeated DNA are separated from bulk genomic DNA as a different peaks during density gradient centrifugation, right? And the bulk DNA forms a major peak, and the other small peaks are referred to as satellite DNA. Now, 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 as you can see, now the satellite DNA is classified into many categories as microsatellite, mini satellite. Okay, these sequences normally do not code for any proteins, but they form a large portion of human genome. But they form a large number of portion of human genome, right? So this sequence show a high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue from an individual show the high degree of polymorphism, they very become useful identification tool in forensic science. So further, as the polymorphisms are inheritable from the parents to children, so the DNA fingerprinting is the basis of paternity testing in the case of disputes as well. Now, as polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. So polymorphism of DNA is the basis of genetic mapping of human genome as well as DNA fingerprinting. It is essential that we understand what DNA polymorphism means in simple words. So polymorphism is a variation at genetic level arises due to mutations. Okay. Okay, so allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant allele at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at higher frequency, it is referred to as DNA polymorphism. Okay. The probability of such variation to be observed in non-coding DNA sequence would be higher as mutation in the sequences may not have an immediate effect in an individual's productive ability. Okay,